In this video, we will discuss about this ALDH4A1 is essential for MPC1 and MPC2 oligomerizations or not. So in order to check that, what it is, they use a BN electrophoresis assay, which can distinguish between signals for the oligomer MPC1 or MPC2 with the monomer with their respective monomer. So first, when they did this regular Western blot, they are able to see that in the case where ALDH4A1 is knocked down, its signal is not there, whereas in the other case where it is reintroduced in the wild type form or in the mutated form, we can see the signals for all the three. But when they did the immunoprecipitation using MPC1s and MPC2-HA, they are able to see that for the MPC1 they are able to see oligomer formations in the case where the wild type ALDH4A1 is reintroduced and monomer signals is decreases compared to the case where ALDH4A1 is knocked down. And similar results they have seen for MPC2 oligomer also, indicating that the ALDH4A1 might be helping in this MPC1 and MPC2 oligomer formations. And we can see that oligomer formations of this MPC1 and the ALDH4A1 is also increased when the wild type ALDH4A1 or ALDH4A1 in the form of mutated ALDH4A1 is introduced compared to the control where the control vector is used. Thereafter, they wanted to check in this panel C, they wanted to check whether this ALDH4A1 actual levels will get affected when the the MPC1 and MPC2 actual levels get affected when the ALDH4A1 is knocked down or reintroduced. So in this case, they can see that its level doesn't get affected much and compared to the controls. And when they did the BN page study to distinguish between oligomer and monomer signals, they are able to see that in the case where ALDH4A1 is knocked down, the oligomer signals goes down and monomer signal increases. Similarly, similar results have been seen for MPC2 also. And when this ALDH4A1 is introduced, they are able to see increase in the oligomers MPC1 and MPC2 oligomer signals, and their respective monomer signals goes down. But and this ALDH4A1 signals both for the oligomer and monomers increases. And thereafter they check the functionality using the pyruvate uptake, and they can see that for the wild type and the mutations, they use different kind of mutations. So for the mutations, except for this 180 to 196 mutations, the other mutation shows a recovery of the pyruvate uptake. But for this ALDH4A1 mutations at around 180 to 196 base pair, they do not see a recovery of the pyruvate uptake. And here is the Western blot result shows the expressions of the ALDH4A1 once it is reintroduced in 3 different conditions. Thereafter, they wanted to check whether this monomer also has an effect on this. Now they wanted to see the effect of these different kind of mutations on this oligomer formations. So here we can see that since for the third case they do not see any pyruvate uptake or effect of the ALDH4A1 reintroductions, similar results they have seen for the Western blot also, BN page also, where for the wild type ALDH4A1 reintroductions and the mutated ALDH4A1 reintroduction, they are able to see oligomer formations for MPC1 and MPC2, but not for the third mutation, not for the third conditions where they use the mutations around 180 to 196 base pair. The range might be wrong, you can check that in the paper whose reference link in the descriptions. And in all the 3 cases we can see the ALDH4A1 formations is there. So this indicates that the third or the second mutations that they used, that the ALDH4A1 mutated around 180 to 196 base pair, does not have proper functions to form a complex with this MPC1 and MPC2. And thereafter they wanted to check the effect of this, whether either MPC1 knockdown or MPC2 knockdown, or both the knockdown has on ALDH4A1 oligomer monomer formations or not. And when they did the BN page analysis, they are able to see that there is no effect on the ALDH4A1 monomer or oligomer formations. And when they just extract, and they are also able to see that in the case where MPC2 is knocked down, they are able to see complex formations for this MPC1 and MPC1 and ALDH4A1, and in the case where MPC1 is knocked down, they are able to see a complex formation of MPC2 and ALDH. In the case where MPC2 is knocked down, they are able to see complex formations of the ALDH4A1 and MPC1, and vice versa in the case where MPC1 is knocked down. And then they wanted to check whether these complex formations will also get affected or in these different conditions or not. And so what they did is they immunoprecipitate ALDH4A1 in the conditions where MPC2 is knocked down, and they are able to see that MPC1's complex formations around 480 kilodalton and ALDH4A1 complex formations around 480 kilodalton, but there is no formations in the MPC2, indicating that the native complex structures are formed even either MPC2 is absent or MPC1 is absent. We can this one is I is for panel I is for the case where MPC1 is absent. They are able to see the complex formations, native structures, native complex formations is around 480 kilodaltons. That is seen for MPC2 and ALDH4A1 signals, but not for MPC1 since it is knocked down. And for the case where MPC2 is knocked down, they are able to see signals formations for MPC1, ALDH4A1, indicating that they have formed negative structures in the absence of either MPC1 and MPC2. But when both are absent, they are able to see non-native complex formations whose size is low. That can be seen in the panel K results, and the ALDH4A1 complex formations can be seen in the lower side. Next, so this is what they did is they check the ALDH4A1 signals in IJK, and during the immunoprecipitations using this MPC1 or during the Western blot in the case where MPC1 is knocked down or MPC2 is knocked down, or both are knocked down. And then they check in the panel L is that in the case where MPC2 is knocked down, when they knock down the ALDH4A1 also, then what they see, they can see that MPC1 signals also goes down. It can be seen here. In this case, in the panel L, only the ALDH4A1 is knocked down, and so they wanted to check whether it will affect the complex formations of the MPC1 or MPC2 or not. They immunoprecipitate the MPC2 signals, and they can see that MPC2 interactions with MPC1 goes down compared to the case of the control. And this is the Western blot for MPC2. Since they are extracted MPC2, its signals remain stable, and the ALDH4A1 signal also goes down since it is knocked down in this case. And then they do this, use the inhibitor that is UK5099 to inhibit the pyruvate import, and in that case they do not see change in the protein levels of either MPC1, MPC2 or 4A1, but when they use the cross linkings, they are able to see the oligomer formations and also slight monomer formations for the MPC1, and similar results they are able to see for MPC2 also, that MPC2 formations are also there when this ALDH4A1 is present or ALDH4A1 is not present. That can be seen here, and though the MPC1 signals are very faint. So this is how they come to a point that ALDH4 is essential for MPC1 and MPC2 oligomerizations. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.